So, can I ask all the speakers to please come up to the and take a chair? Just trying to keep you active. And could those people who have questions to ask, please make yourself visible to those with microphones?
Yes, a question about the polygenic risk factor. It's a scientific question. There is an argument whether, there are, I don't know, let's say 100 or something like that genes where there is a mechanistic connection to the phenotype, or is it really relevant to have 10,000 variants that are statistically appear? In the data you presented, is there a big difference in the performance and the really ability to separate the high risk from the low risk if you use thousands of loci or only hundreds?
That's a good question. The short answer is yes. Going from the top 100 or few hundred loci to tens or hundreds of thousands of variants increases predictive performance, I want to say significantly, but non-trivially. So I can't remember the exact numbers, but with our data, if you just used a set of 100 or 200 loci for coronary disease, maybe relative risks are 3 or 3.5, and then if you use many, many more variants across the genome, those relative risks go up to, in an extreme tail, go up to 4.5 or 5. So it does make a big difference to include lots of the genome for prediction. Of course, there's a separate question about using these sorts of resources to understand biology, where I think that the loci where you're clear about what's happening are much more helpful.
Peter, can I ask a question to add on to that? In our hands, when examining polygenic risk scores in non-European populations, we've not necessarily found the same to be true, and that using just the sort of genome-wide significant or a smaller set of variants might suffer less from in the translation to other populations. Have you seen the same thing?
Absolutely, yep.
Hi, this is a question mainly for Jonathan. So to what extent, your numbers when you're looking at loss of function mutations and how many genes have loss of function mutations, to what extent did you filter those variants for things like end truncations that don't undergo nonsense-mediated decay, or variants that are only on exons that aren't actually expressed? Because when we look in NOMAD, those numbers are actually a lot lower when we do any kind of filtering on those variants.
Yeah, I totally agree. It really does matter. I think the numbers in NOMAD are lower. I think one of the reasons, we looked into this several reasons. The precise definition of what counts as a loss of function variant matters. The filtering in terms of their how deleterious they are, and also indels. So one thing that we include are indels, I think, NOMAD less so, and that does change the numbers a lot. So I think this is a conversation that the community has to have. Is there really one canonical definition of a loss of function? Probably not. It's a sliding scale. Whether you include indels or not makes a big difference.
I've got another question for Peter about the polygenic risk scores and the low correlation with family history. I mean, there are several possible contributing factors to that. I was wondering if you could just talk briefly about those and what you think is going on, and also to what extent this differs between different diseases.
Good question. So we don't know the answer, but natural hypotheses would be, and probably many of them, family history is probably capturing to some extent shared environment as well as shared genetics. Family history, so close relatives affected by disease, will upweight the effects of rare variants in a way that polygenic risk scores won't. And polygenic risk scores, certainly for some traits, may be we would hope less for disease traits, but could be capturing things other than kind of direct genetic causes. They could be surrogates for social, socioeconomic markers, or nurturing variants and so on, where family history would be less relevant. There's also the kind of obvious thing that polygenic risk scores are sort of less heritable than rare variants. So if I'm in the top 1% polygenic risk score for a particular disease, the chance that one of my first degree relatives it's 10%, it's not 0, but it's not massive.
Okay, we'll go to the back.
Thank you. Nick Harvey from the MRC Life Course Epidemiology Unit in Southampton. First of all, thank you very much to all for fantastic talks. My question was really around the polygenic risk scores again, so I guess directed at Peter. We've been doing this sort of thing for bone outcomes with others. And I was intrigued by the almost complete independence of the PRS from the Q risk. And I just wondered what considerations there were in terms of the mechanisms identified by the PRS relative to the treatment you might give, so how it might be used in clinical practice. And I wondered whether the sort of statin pathway or the cholesterol side of things was captured by the PRS, and whether people identified in that way might be the best people to treat with statins, or whether they are best identified through the Q risk, and how those things sort of come together in that sort of distinction between maybe a relative risk reduction, but at the lower end of absolute risk, versus a reduction at the higher end of absolute risk.
Yeah, it's a great question. So to put it another way, how do we know that if you have a high polygenic risk score, but not necessarily high traditional risk scores, statins would be a good thing?
Absolutely.
There's pretty encouraging data on that. So Sate Katharesan has a really nice paper where he went back into the many of the large statin trials, and where there was genetic data available, he broke the individuals in the trials up into 5 groups, the bottom 20% of PRS, the next, and so on. And that study showed that amongst those groups, it was the top 20% where the effect of statins was largest in both relative and absolute risk reduction. So that's at least encouraging evidence that those groups would benefit from statins.
Thank you.
So number 2, have you got another?
Thank you very much for the great talks. That's Joanna Julaki from Imperial. I will follow up on the polygenic risk score. I take the point that it's independent from Q risk, but at the same time, the data you showed clearly that the absolute risk difference in the groups is not higher. In other ways, the people that have Q risk below the threshold, despite the fact that their polygenic risk score was high, they were still below the threshold for treatment. So in a way, the data is showing that the polygenic risk score doesn't add to the predictability of the Q risk.
So I may misunderstand the question, but I think it's clear from our analyses, and others have done similar things, that if you combine polygenic risk scores with traditional risk factors, you get improved risk prediction. So you get more people beyond thresholds. In some sense, you're spreading out your estimates of risk because you can do that more precisely, and you get different people beyond the threshold. So there are individuals, were you to think that the threshold of 10% risk over 10 years is in some sense an important threshold, and certainly the UK guidelines for statin recommendations, if you think that's an important threshold, then there are, in our projections from UK Biobank to the UK population, as I said, there are sort of 500,000 people who actually have that level of risk that you're not aware of if you just use the traditional risk factors.
Maybe it was not on the slide.
We have actually done the same thing in Iceland, looked at the polygenic score combined with the traditional risk factor, and we get out with exactly the same results as Peter.
Yeah,
Okay.
Thank you.
Okay, we'll go to number 1, yes.
Hi, I'm Jackie McCarthy from EBI. I've got a question for Carrie. When it comes to whole genome sequencing of UK Biobank, I was wondering what strategy you would recommend for sequencing this resource, like short read sequencing versus long read sequencing, based on the number of individuals and the costs involved, and obviously the advantages when it comes to long read sequencing and the interpretation of structural variation.
We have sequenced, whole genome sequenced about a little bit over 80,000 people in Iceland with short read sequencing, Illumina technology. We have sequenced about 2,000 people with Oxford nanopore technology. And we are basically forced to use the short-read technology as sort of your basic sequencing because the error rate is still too high. The data management coming out of the ONT technology is too difficult still to use it as the sole technology, but there is no question about it that the reason we are sequencing with ONT technology is that it gives us much, much more sensitivity when it comes to picking up structural variants, and we pick up probably about 3 times more structural variants using the ONT than a short-read technology. So if there was no resource limitation, my recommendation would be to use the short-read sequencing on all of the samples and then sequence a subset of it with ONT technology, something like 10 or 20,000. But as I said, that is the desire of an old man who sees no resource limitations anywhere where he goes.
Thanks. Okay, I think there's another one down here.
Question for David Meltzer. Have you done the GWAS for penetrance of C2H2Y?
We're currently doing that in collaboration with the US study, the HERS study, and we've approached a French group. We've got some encouraging data, but we need to replicate it first before talking about it.
And I think there's, yes, over there, number 1.
I was interested in the announcement from John Bell on the early diagnosis initiative and just wanted further details on whether that was based around a research opportunity or principally around translation or both.
Yeah, so it's going to be a research study, but it's going to be, has to by definition be quite closely integrated with the NHS. So we'll be migrating data back to patients and into the healthcare system. So it sort of sits between the two. Obviously, we can't recruit those numbers of patients without using NHS systems, and we will ultimately, I think, have a duty of care to return data to patients. So by definition, there will have to be return of data in a variety of settings. But that obviously there's a whole set of issues because it's a new type of cohort where there will be a dialogue between the study and the patients. But that's broadly what we're going to try and do.
Okay, I think we'll stop there because we have a fairly tight time scale for lunch. Lunch will be served upstairs where coffee was on arrival. You have exactly 1 hour, and I'm being told exactly 1 hour to eat. So on your marks, get set. And let's just thank the speakers from all of this morning's session again.